Many traits do not follow Mendelian predicted ratios, including linked traits. We start thinking about Mendelian, again, being able to have mental representations of what to expect. So if I say dihybrid F2 phenotypic probabilities, that's a lot. We have two traits. We have to understand that the F2 generation is the result of crossing two heterozygous F1s, and phenotypic probability is what the overall ratio is going to look like. So if you were thinking it would be a 9 to 3 to 3 to 1 ratio, you're on track. Now, how do we get there? We start with P, and when we're talking Mendelian, we say these are non-linked, which means that the gene for the L trait and the B trait are on separate chromosomes. So in this, in the P, we have the purebred dominant and purebred recessive, and in F1, we're going to end up with this heterozygous genotype. When we cross the two heterozygous genotypes in the F1 generation, we end up with this 9 to 3 to 3 to 1 phenotypic ratio in the F2. Now, sometimes genes can be linked. So in this case, the traits are on the same chromosome. So now this acts much more like monohybrid crosses. So when we are looking at this, it's still dihybrid, and we still have this P generation where we're purebred and purebred, dominant and recessive, and we still get this F1 generation that is heterozygous. And when you cross them up, you end up with an F2, but that F2 has a 3 to 1 phenotypic ratio, not a 9 to 3 to 3 to 1. So when we look at F2 probabilities, again, if we're thinking Mendelian, non-linked, you're going to have all of these possible gametes from these heterozygous, and you're going to have on the same side, other side, again, the same idea of all these different gamete combinations, and you end up with 9 to 3 to 3 to 1. When they are linked, however, we end up with only two possibilities. It's either this chromosome or it's that chromosome. Same thing on the other side. And now we're not looking at a big 4x4 Punnett square. It's really a 2x2. And when we put these together, when we're talking phenotypes, we're going to get a 3 to 1 phenotypic ratio. And we're not even going to get these other varieties that could have been possible with Mendelian non-linked traits. So again, when we're thinking this, Mendelian non-linked is 9 to 3 to 3 to 1. But for dihybrid F2 probabilities with non-Mendelian linked traits, linked genes, we're thinking 3 to 1. Now, when we start getting into this, we also, with linked traits, we also get into crossing over. So linked genes have a recombination frequency of less than 50%. So as we start looking at this, again, during synapsis, during prophase 1 of meiosis, we can have this crossing over take place. And if the genes are really close, they would like only 5 map units apart. That 5 map units comes from the chance of recombining these genes are only 5%. However, if these genes for these traits are a little bit further apart, now we can get some new combinations of the alleles combining. So if on average, if they would have a 30% recombination frequency, we would say they are 30 map units apart. Again, this idea that crossing over doesn't create new alleles, it just recombines the existing alleles in new ways. So we can get some new recombinations the further apart they are. So again, when we start looking at this, we can also look at test crosses. So non-linked test crosses, if we had this F1 heterozygous crossed with the homozygous recessive, we would end up with these possible gametes, that possible gamete, and these phenotypes expected for Mendelian test crosses. However, when we start looking at linked non-Mendelian test crosses, we still have dihybrid, we still have the same genotypes. However, when these traits are linked, we are only going to end up with these two possibilities, where because the L and the G are on the same chromosome and this recessive L and G are on the same chromosome. So we only end up with two possible phenotypes possible in as expected for linked test crosses. Again, sometimes we end up with unexpected ratios. And when the ratios don't turn out the way we expect with Mendelian, we can use chi-square analysis and different things like that to see if they fall within a statistical range of what's expected. But again, this is just another way to visualize what would be happening. So again, when we have these P generations, if they're non-linked dihybrids, we would end up with this heterozygous for our F1. However, when they're linked on the same chromosomes, we would expect this arrangement for our heterozygous F1. So then when we put these heterozygous F1s together with a test cross with homozygous recessive, we would expect this 1-to-1 to 1-to-1 ratio where we get all of the different phenotypes in equal proportion. On this side, we would expect with this test cross to end up with like a 50-50 combination of phenotypes. However, sometimes things come back unexpected. You would only expect these two phenotypes to show up, but every once in a while, you can still get some of these other alleles. And what in the world would be the cause of that? And it comes back to crossing over again. Again, during synapsis, we can swap some of these genes to create new combinations of alleles. So again, this idea, we are being able to, if this is a dominant, like big G, big D, through this crossing over, we can end up with a big G and a little d, and a little g and a big D here. So then we brought back these different recombinations or new combinations of alleles that already exist, and that's where we get our recombinant and slightly different expected ratios. Many traits do not follow Mendelian predicted ratios, including linked traits.